So the last part is about the connectivity map and this idea of the links, and I'll show you how this idea of treating human cells with a whole bunch of drugs and then measuring gene expression before and after is very useful for drug discovery. It's an alternative approach to some of the approaches that you were introduced in this class so far. This started in this paper from 2005 or 6, where again this group at the Broad, they call the connectivity map group. They use gene expression microarrays to measure the effect of all of the FDA-approved drugs treated with on 4 human cell lines before and after, and they had 7,000 microarrays that were done on. So this was a big project, and at that time when it was done, and then we created this database called the Connectivity Map Database. And the idea is that you can now come to this database with your own data, where you either have a mouse model of disease, you measure some small molecule in human cells before and after perturbation, or you actually have like normal tissue versus disease tissue, and you want to find small molecules that would either reverse or mimic your differential expression. So you compute differential expression, you have up and down genes, you query those genes against this database, and then you get positive and negative connections. So this was sort of like a big transformative paper, and the NIH wanted to expand this project, and they challenged and other people to come up with cost-effective ways that this could be scaled up. So basically the original study only did FDA-approved drugs, but what about doing it for thousands of small molecules, more cell lines, more concentration? Can you do it at lower cost? So the people at the Broad came up with this L1000 technology. It's a Luminex bead technology where they measure about 1,000 genes, and then they pick those genes so maximally to be able to infer the expression of the rest of the genome. So they only measure 1,000 genes, but those 1,000 genes are representative genes. They're called the landmark genes. That's why the L is from landmark. And then using a linear method, they can infer the rest of the genes. And it's they show that it's as accurate as microarray. So in the initial phase, the data is available on this clue.io as well as Geo if you want to play with it. Over 20,000 small molecules. They knocked down with shRNA all of the genes, and 77 cell lines. Mostly, there's a core of about 10 cancer cell lines, and then in total they've done over 1.4 million profiles and about 500,000 signatures. That means that they did everything in quadruplicates, so you have repeats for each condition, and then you can compute before and after with those. And they published this in Cell last year, actually a little bit over a year now. It's like a year and 3 months. And they visualized the subset of the data using this Disney plot, where they consolidate the drugs into, regardless of what cell lines they are, and then they also divided all the data set into a discovery set and a touchstone set, and we created this app called L1000 Fireworks Display. And in this plot you see 17,000 points, but here every point is a signature. So a signature, it means that it's a specific drug, specific cell lines, concentration, and gene expression was measured at a specific time point. And then the coloring is done by the known mechanisms of action of the drugs. Okay? So this green sort of a cluster is HDAC inhibitors. So you can see in this map, you only get dark green in this cluster. And it's very concentrated with HDAC inhibitors. The good news here is that those gray nodes, like it's a little bit, doesn't look that good, but if you go to the paper or the website, it looks better. But those gray nodes are small molecules that until now nobody knew what they do. So those are predictions, or it's pretty clear just by eye that those are HDAC inhibitors also, or very likely to be HDAC inhibitors. And then you can do other assays to validate whether those are HDAC inhibitors. But then you have also those other types of drugs. So here is topoisomerase inhibitors and CDK inhibitors that follow this cluster. And then here you have this glucocorticoid receptors. So for many, I guess, typical FDA-approved drugs that exist, you can immediately discover new small molecules that you can pursue. So that's pretty exciting.